good public structure for kind of keeping that together. But I think there's two things feeding into why cancer looks so much lower here. I think one, you're absolutely right. There may have been some underdiagnosis at that time, and people who had cancer and died, and it wasn't appreciated that they had died of cancer. The other thing is maybe that cancer is a disease of age in many cases. And so the longer you live and the longer the lifespan is in a specific kind of country or region, then the more deaths that we often see attributable to cancer. Does that make sense?
Thank you. Appreciate that.
Mm-hmm. Okay, so TB today is a major cause of death worldwide. And so what I'm showing here on the left, this little bar graph here, can you see my pointer? Yeah, good. Okay, so this is the top 10 causes of death in low-income countries. And so what we see here looks a lot more like the picture that we saw in 1900 in the United States, where you see lower respiratory infections. Here we have malaria, tuberculosis, HIV, all these infectious diseases as leading causes of death. And so TB, this is actually a little bit outdated. And TB is now, other than COVID-19 for the last couple of years, TB is now the leading cause of death in terms of a single infectious disease agent worldwide. And so it's estimated that there are about 2 billion people that are infected with TB worldwide. There are about 10 million new cases every year, and somewhere between 1.5 and 2 million deaths are attributable to TB worldwide every year. And the reason I think that many of us don't really think about TB that often is because this is a map on the right that's showing the global burden of cases. So where do the most cases happen? And you can see that here in North, Central, and even South America, we have relatively little tuberculosis. And that's also true here in Western Europe, Northern Africa, and much of the Eastern Europe and the Soviet Union, although there is actually plenty of.
Sarah, there's a question.
Yeah.
It said, isn't TB curable or treatable now?
Yes, I'm going to get back to that in just a minute. Thank you, Susan. And thank you, Sanchita. Okay, so where TB really is happening is in sub-Saharan Africa. That has the highest incidence of TB in the world. So that means the largest kind of percentage of the population has TB. And then there's a lot of TB in Southeast Asia, in particular, in South Asia, in particular, in India has a really high burden of TB, as well as Indonesia, and lots of cases in China, as well as other parts of East Asia. So why is TB, I think this starts to get at Susan's question, why is TB such a challenge today? I think that many of us are kind of accustomed to thinking of, viruses are the real threat because we don't have that many great antivirals and we have a lot of antibiotics. And so, bacteria are a problem that's been solved. But that for tuberculosis, that's particularly not true. I think that's also not true for many bacterial pathogens, given the rise of drug resistance. This is a little bit of an aside, but the CDC has estimated that if the world doesn't change the way they approach kind of finding new antibiotics, which doesn't really happen that often anymore, then it's estimated that by 2050, that infectious diseases will again kill as many people as cancer does. So bacterial infections in general are a major problem, but TB in particular is very difficult. And one of the reasons is because once you're infected with TB, you're infected for life. It's thought that the immune system is unable to clear infection. There is no effective vaccine for TB. HIV and TB are really a deadly kind of synergy that happens in many parts of the world, where HIV makes TB worse and TB makes HIV worse. So TB is very difficult to treat with antibiotics, and I'll get into that in just a few slides. And as a result, drug resistance is really on the rise. So this is now a map looking at the emergence of drug-resistant TB. And so again, you can see that there are many places in the world where there are real hotspots of drug resistance, although those don't necessarily correlate with where we see the highest burden. So the way that you can interpret this map is going from this kind of nice green, I don't know what shade of green this is here, emerald green here, all the way down through red is the lowest to the highest amount of drug resistance. And so there's parts of the world that have really, really high rates of drug resistance, including parts of the former Soviet Union, as well as some parts of South America, China, and also some of East Africa. But worldwide, it's estimated that, this is multi-drug resistant TB in this map. So worldwide, it's estimated that around 20% of all TB cases are resistant to at least one drug, 3.7% of all new TB cases are multi-drug resistant, and 20% of previously treated TB cases are multi-drug resistant TB. Okay, so in the context of TB, I should explain what we mean by multi-drug resistant. So unlike if you were to develop an infection and go to the physician, like a urinary tract infection, let's say, and go to the physician, likely you would be treated with a short course of one antibiotic, that you might take for a week or two. If you're diagnosed with TB, then if you have drug-sensitive TB, you have to take a cocktail of 4 antibiotics for a period of 6 months in order to completely eradicate infection. And so what that means is that that these drugs are also very toxic. They make people feel sick. And so what that means is that a lot of people start their therapy, and as soon as they start to feel better, they stop taking their medication. And I think we all know, like, that's one of the things that physicians tell you, do not do that, right? Because it allows drug resistance to develop. And so what multi-drug resistant are resistant to the two best drugs in that 4-drug regimen. Did I hear another question?
No, I have a question, sorry, Sarah. So I'm from India. We all received vaccine when we were young. So those vaccines are not effective. That's what you're saying?
Yeah. So this is the vaccine that you receive. It's called BCG. So BCG is given in pretty much every part of the world where there's a high number of cases of TB. BCG was first introduced around 100 years ago. It was developed at the Pasteur Institute in France, and it is one of the safest and most commonly administered vaccines. So the reason that people give BCG is because it has clear efficacy against childhood tuberculosis. So the vast majority of cases of TB are in adults, but there are a smaller number of cases that happen in kids, and in kids it can be really severe, disseminated throughout their body, and it can be very hard to diagnose in kids. And so if you give BCG, you can prevent those serious cases in children, but you don't prevent adult pulmonary disease, and so you don't really make an impact on the epidemic itself. If that makes sense.
Got it.
Because the efficacy of BCG wanes for reasons that we don't understand. So it seems to protect people for about the first 10 years of life. And then even if we give a booster in the same way that, we give boosters for COVID-19 or for, hepatitis or other vaccines, it doesn't work. And that's something we really don't understand. And that's one of the reasons why TB is still so widespread, despite the fact that there is a vaccine that is widely given. So one other thing to say about kind of the tools that we have for TB is that if you look at drugs that are used for treating TB, this is when they were discovered, okay? So there was a new one in the last couple of years, so I should add something here. But for the most part, all of the drugs that are efficacious that we use for treating TB were all discovered decades ago. And so the tools that we're using are these drugs that were discovered decades ago, this vaccine that doesn't really work that was discovered 100 years ago. And so this is one of the reasons why TB is still a major public health crisis that, causes a lot of deaths every year in the world. So one more thing I'll mention is before kind of getting into the biology, and is that it's also really hard to work on TB. So TB is, because TB if you get infected with TB, I'll talk about this in a moment. If you get drug-resistant TB, it's very difficult to treat. Even if you get just drug-sensitive TB. The drugs that you use to treat TB are quite toxic, and so some people have a difficult time tolerating them. And so TB is considered to be a dangerous pathogen that's been classified as a biosafety level 3 pathogen. So what this means is that if you want to work with TB in the lab, then you have to work in what's called a biosafety level 3 facility. And I know all of you who've worked with me on this call in the past know something probably about BL3, or at least know how much money I spend in my research. So BSL3, we have 2 BSL3s at Berkeley. They are primarily facilities where we work with TB, although we did also work with COVID-19 in those facilities. But the BSL3 facility is a special suite of rooms where the airflow is controlled, and all of the air movement we pull air in from outside the BL3 into the facility. So the reason for that is if there's a spill inside the facility, it can't escape to the outside world. Everything we do with live TB, we do inside a biosafety cabinet. We never take anything out of the biosafety cabinet without sterilizing it first. That includes our hands. And so it's a really unusual way of working in the lab. It's different from the way, most biologists work in the lab. We wear this protective equipment. We wear these Tyvek suits. We wear this is not a picture of our BL3 actually, but we wear what's called a PAPR, which you've probably seen in the context of COVID-19. It looks like a space helmet. And what it does is it blows a curtain of purified air over your face so that you can't get exposed to the bacteria. So you have to have a BSL3, you have to have this infrastructure for working with TB. And then if you think about all the things that the PIs here need to buy, like microscopes or other types of instrumentation, animals, all of this has to happen for us within this highly specialized facility. And so TB is more difficult, and it's also a lot more expensive to do the work. So I think this is one of the reasons also why, even though we have a really robust group of PIs who work on TB at Berkeley, me, Jeff Cox, Russell Vance, and then Lee Riley, who passed away last fall, but there aren't very many people studying TB around the world, even when you compare to another infectious disease like HIV. Okay. So I'll tell you, are there any questions about I'm going to move on from kind of the public health aspects of TB and get into the biology, unless there's any questions. Okay. So like COVID-19, TB is a disease of the lungs. And so, what happens when a person has active TB is that, they have bacteria growing in their lungs. Then they cough, they cough out aerosols that contain the bacteria, and these are breathed in by, an uninfected individual, and then that person can become infected. So what happens when TB enters your body is that it is taken up by these cells called macrophages. So this is where TB is a little different from COVID-19. COVID-19 infects the actual cells of your lung, the parts of your lung that do oxygen exchange. Those are the cells that COVID-19 infects. TB is a little different. It infects macrophages, which are cells of the immune system. And macrophages, and I'll tell you a little bit more about this in a moment, macrophages, their job is to uptake organisms, bacteria, fungi, other organisms like that, and kill them. That's their job. But what TB does is TB comes in and it makes a home in these macrophages. So in the beginning, TB replicates in these immune cells in your lungs. What happens over time is that new and different immune cells come in to try to fight the infection. And so maybe you've heard of CD4 T cells, because these are the cells that get infected with HIV and are killed by HIV. So CD4 T cells, they are kind of the master regulators, or sometimes people call them helpers of the immune system. What they do is they come in and they instruct other immune cells to really make a stronger response. So what happens is that when CD4 T cells that recognize TB come into the lungs, they tell the macrophages, okay, it's okay. You can do absolutely your max to try to kill the bacteria. And so then what happens is there's some control of the bacterial infection. Bacterial numbers go down. But what happens is that this immune cell interaction is not sufficient to eliminate the infection. And so people who have been infected with TB have what we call latent disease for their entire lives. And so what that means is that they may not have symptoms, but the bacteria are in there somewhere. And this, these two cell types, CD4 T cells and macrophages, are kind of working together to fight the infection. And if this kind of immune control ever wanes for any reason, so that might be pregnancy or old age or malnutrition or, something that causes your immune system to be a little less strong for a period of time, then what happens is the bacteria will start growing again. And your immune system kind of loses the ability to control the infection. You get a lot of bacteria growing inside macrophages in your lungs. What eventually happens, it's very similar to COVID-19. If you remember that during COVID-19, the thing that killed many patients is the immune system becomes too strong. And maybe you've heard the word cytokine storm, that can lead to the shutdown of the lungs and the death of the patient. So a very similar thing happens in TB, where when the immune system is unable to kill the bacteria, more and more immune cells just keep coming in. They keep trying to kill the bacteria. They can't really kill the bacteria. Instead, they kill the your own lung tissue, and then your lungs are unable to infection, and then you succumb to disease. Okay, did that make sense? Okay, great.
There's a question.
Oh, yeah. Are people contagious during the latent period? No, for the most part, no. I mean, essentially, no, people are not contagious. Really, people are contagious with the TB once they have symptoms. So that means once they're coughing. There's a couple reasons for that. So during latency, it's thought that this immune structure here that I didn't mention, it's thought that TB is contained within these immune structures that are walled off from the rest of the body. So TB is in these macrophages, and then you build this layer of of fibrous tissue that makes like a capsule around this thing. And then there are T cells around that are kind of maintaining the macrophages so that they're activated and they're controlling the infection. And so the idea here is that now TB is trapped within this granuloma and it can't spread from, to a new place. When you have active disease, this structure breaks down. You have bacteria growing not just in macrophages, but bacteria start to grow extra outside of cells and just kind of in your tissue. You get tissue destruction, you cough, and then that's when you spread. Yeah, good question.
Where do the granulomas hang out?
The granulomas are in your lung tissue. So, you can't see this at all, but if you can kind of like peer at this teeny little lung, and I've just kind of drawn these little green circles everywhere. So just throughout your lung tissue, you'll have these little pockets of granulomas in them. And then depending on the patient, you might have a lot of them or you might have few of them. Yeah, so they're in your lung, but they're just, as long as you're not having a lot of bacterial replication, so you're not having a really strong immune response, you just don't even know they're there. So you have no symptoms.
Can you see those on an X-ray or other kind of imaging?
So what you see on an X-ray is if the granuloma has grown big enough so that it's made a hole in your lung tissue. That's called a cavitation. You can see that. There are granulomas that probably have active, that have bacteria in them that you can't see on an X-ray, but what you can see them on is on a CAT scan. And then especially if you combine that with PET, then you can actually see. One of the reasons we know that there's stuff going on in these people who have, latent disease is that if you do a PET CT, you can actually see hot spots in their lungs where the bacteria are there. Okay, so I think in the interest of time, I may skip that and then talk a little bit more about macrophages specifically. So macrophages are my favorite cell type. This is one of those things that probably seems really weird to non-scientists. I know, to my family members, it definitely seems weird, but I've studied macrophages for, 23 years. And I still don't know anything about them. So which is crazy. So macrophages are the main cell type in your body that kind of surveys all around the body, identifies when there's a pathogen that shouldn't be there. And then when it identifies a pathogen that shouldn't be there, what it does is it engulfs it through this process that we call phagocytosis. So here's an illustration of a macrophage. So this is like just a part of a macrophage. So the yellow part is inside the cell, the white part is outside the cell. A macrophage will grab onto things like a bacterium or a yeast, and it will kind of pull them into the cell so that now they're inside the cell, inside this vacuole. And then what the macrophage does is it just bombards that vacuole with all kinds of things that are toxic. So I'm going to actually start with this one. Oops. Oh no, is it not? Okay, just give me 1 second because I really want to show you this. Okay, so one of the things that macrophages, oh no, it's not working here either. Darn. Okay, well, I'm not going to be able to show you that movie, but one of the things that macrophages do is, if you get a cut or an infection on your skin at home and you buy hydrogen peroxide and you can put it on the cut and you see this like bubbling and that kills the infection. Macrophages actually make hydrogen peroxide inside of the cell, and they make the hydrogen peroxide so it's concentrated right in that vacuole where bacteria or something that's infected you is. And so your body makes that hydrogen peroxide and puts it in there, and that can lead to killing of most microbes. Your macrophages also make bleach, the same chemical that's in bleach, and make it in a really localized way and put it into this phagosome as well. The other thing that happens is, just like you have enzymes in your stomach that digest food, there are enzymes inside of macrophages that are digestive enzymes that are also sent into this vacuole where pathogens are. And so these digestive enzymes can also work on digesting what's inside the phagosome. There are also ways that the macrophages try to starve a pathogen that they engulf. So they will, all cells need metal, right, to function. And so macrophages will just suck up all the metal, take it away from where pathogens are able to access them. So, macrophages and then their kind of sister cells, neutrophils, they have all these ways of killing pathogens. But one of the things that is unique about TB is that TB is resistant to all of these mechanisms. So this is a piece that was written by my friend and collaborator, Heron Darwin. Honey, if you're here, now you'll get some context for Heron. But Heron is a colleague of mine. She's a collaborator. She works on the project I'm going to talk about today. And she wrote this piece titled Mycobacterium tuberculosis, the honey badger of pathogens. And the reason that, she called it the honey badger, if you know anything about the honey badger, maybe that's a link to a video of a honey badger I hope it is, in the chat. But TB is just resistant to everything that I just told you about that macrophages use to kill microbes. TB is resistant to those things. So TB is highly resistant to hydrogen peroxide. It's resistant to all of the digestive enzymes that try to degrade bacteria. And so this is one of the reasons it's thought that people once you get infected with TB, it's really hard to ever get rid of the infection. Okay, so what I want to, that's a video of a macrophage. That's cool. Yes. And if anyone's interested, I have some really, really cool videos as well that I can share offline. But so what I want to talk about today is, I just told you there's all these ways that we know about that macrophages can kill bacteria, but those things don't seem to work with TB. And so one of the questions that my lab focuses on is, how do macrophages contain infection with TB? How do they kill them? Because we know that they can effectively kill them, but we just don't know how. We don't know what mechanisms they're using. So maybe you're asking yourself, like, why would you want to know this? Really, what you need is a drug, right? Or you want a vaccine. And my lab does work on those things too. But one of the things that I don't have a slide about, but that I wanted, an idea that I kind of wanted to get across, is that a lot of times in infectious disease, it seems like your immune response is working really well because you have a lot of inflammation. But what's actually happening is that you have a lot of immune cells that have come to a tissue, and they're doing their best to try to control the infection. But the actual cell that needs to do the important work of killing the pathogen has been impaired in that function. So for example, in COVID-19, you might get, the people who die from the disease. I mentioned, and I think you probably already knew, that a lot of times that's because the immune response to the infection is so overwhelming that it kind of ends up destroying the host tissue. But why isn't it killing the virus? Well, one of the reasons is because the virus is able to, in the cells that it's infecting, block everything and kind of hide in that cell, right? And so the immune system is bringing all these cells and making all this inflammation, but it's not having an effect on the cell that's infected. And that's what we think is happening with TB, is that in active disease, you get all of this inflammation, all of these immune cells coming to the lungs, and they're trying to make the macrophages kill the TB, but the macrophages are impaired in being able to kill the TB. And so the immune system just keeps ramping up and up and up, and so ends up destroying the host tissue before the macrophages actually activate to kill the TB. And so one of the kind of new concepts in developing new types of drugs for bacterial infection is to say, okay, if the problem is that the macrophage is not actually killing the TB, can we make a drug that's going to overcome that and allow the macrophage to kill the TB? And then maybe we won't have as much drug resistance because it's not directly killing the bacteria. Okay, so how much time do I have left?
We have until 1 booked.
Oh, okay.
And it sounds, we're answering questions throughout, so we don't necessarily need to leave a big chunk of time at the end.
Okay. All right, so I'm going to tell you about a collaborative project, and I'm showing you the people who are doing this work together with me. Some of you may have interacted with some of these people over the years. So this is me and my postdoc, Sam. This is my collaborator and very good friend, Heron Darwin at NYU, her graduate student, Gina. And then my long-term mentor over, the last 20 years, Dan Portnoy, who's also here at Cal, and we have a shared graduate student, Andrea. And then we have a collaborator in Australia named Sarah also, which gets to be kind of confusing sometimes, but she is an epidemiologist. Okay, so I hope I can just give you kind of the highlights of this story that we've been developing as a group over the last few years. And this story really starts with this observation of the most common mutation that's found in human populations, and which is mutation in this gene aldehyde dehydrogenase 2, ALDH2. And so what I'm showing you here is a map of where this mutation, which causes a non-functional version of this ALDH2 protein to be made, where this mutation is highly prevalent. So yellow is what we would call wild type, so that these people would have a functional ALDH2, and the green is a mutant ALDH2, so a non-functional ALDH2. And what you can see here is that in East Asia, there is a high prevalence of mutations which result in a loss of function of this ALDH2 enzyme. And the reason this is so interesting is because it's not really clear what has been the force that has driven selection of this ALDH2 mutation in the human population, because it is known that people with this mutation have a higher rates of cardiovascular disease, and they have higher rates of digestive system cancers. So there are some very clear epidemiological associations that are negative with this mutation, and it's really unclear what is the kind of benefit that's associated with this mutation. So I think probably all of you have heard of this mutation, even if you don't know the name of the gene that causes it, because ALDH2 is important for detoxifying ethanol. So when we drink alcohol, of course, that has to be detoxified by our body. And the way that this happens is first with alcohol dehydrogenase converts the ethanol that we drink into this molecule called acetaldehyde. And then acetaldehyde is converted by this enzyme that I told you about, ALDH2, into acetate. And acetate is harmless. Acetaldehyde is not harmless, it's toxic. Aldehydes are a class of molecule in general that are toxic. And so people who have this mutation in ALDH2, one of the kind of characteristics is associated with this mutation is that they're unable to detoxify ethanol when they drink it. And so when a person with this mutation consumes alcohol, it causes this acetaldehyde to build up, and this aldehyde causes changes to your physiology, that are negative and that that happen throughout your body, and also help, cause a really terrible hangover in people with this mutation. But one of the things that you can see is that the people, there's effects on the vasculature that causes characteristic flush, called alcohol flushing syndrome. Also, I think a lot of people know this as Asian glow. So probably most of us know people who have this mutation that when they consume alcohol, if they do consume alcohol, they get a really, like a red or a purple face. And that's because of this buildup of this toxic product, acetaldehyde. Okay. So why am I telling you about this when I've just, spent most of this time introducing you to TB? So many years ago now, 5 or 6 years ago now, when Heron was on sabbatical here at Berkeley, she's Korean, and I'm Korean, and we spent a lot of time together talking about science and also consuming alcohol and talking about science at the same time. And although we both have family members who have this mutation, neither of us has this mutation. So we came across this paper in a Korean epidemiology journal that was looking at this mutation and trying to say, okay, so what other diseases are influenced by this mutation? And I have no idea why they looked at these diseases, but they looked at hepatitis, asthma, type 2 diabetes, alcoholism, and TB. So it was already known that if you have this mutation, you're much less likely to become an alcoholic. And that's because most people with this mutation don't drink very often because they feel sick when they drink. And so this is looking at what's called an odds ratio. If you're below 1, that means that you're less likely to have the disease. Above 1 is more likely to have the disease. So you can see here that alcoholism, people with this mutation are protected against alcoholism. But the surprising thing here was that they looked at TB, and what they found is that people with this mutation were also protected against having active tuberculosis. So what the people who wrote this paper proposed was that if you have this mutation, you're less likely to drink. If you're less likely to drink, then you're more likely to have a healthy immune system. If you have a healthy immune system, you're more able to contain TB infection, and you don't transition into that phase of active disease. But I'm going to skip that we reproduced this in a cohort in Vietnam with our collaborator, Sarah. But what Heron and I got really interested in is that we thought, okay, we know that this gene ALDH2 detoxifies aldehydes. And the other thing that we know is that in macrophages, when they get activated, there's all of these changes to their metabolism. And, the specific details of that are not that important. But aldehydes actually make, macrophages actually make a lot of aldehyde when they get infected and when they get activated. And everyone's always thought of this as kind of like a byproduct of things they need to do. And so they just have to detoxify this aldehyde. So one of the aldehydes that macrophages make is formaldehyde. So you've probably heard of formaldehyde, like if you remember dissecting something in biology class in like 7th grade and the animals are preserved in formaldehyde, or if you, like me, before I found out what it was, if you used to go get, like a Brazilian blowout for your hair, the straightening treatment, that what they use is actually formaldehyde, which is terrible. Even the ones that say they don't use formaldehyde, they do use formaldehyde. So I have stopped doing that. But cells also make formaldehyde as a byproduct of metabolism. And the enzyme that detoxifies formaldehyde in macrophages and other cells, as well as other toxic aldehydes that are made, is ALDH2. So what Heron and I, what we hypothesize is that in humans who lack a functional version of ALDH2, when they get infected, their macrophages will make a lot of aldehyde. This aldehyde is very toxic. They're unable to detoxify this aldehyde, and that aldehyde actually kills bacteria. Does that make sense? Okay. So I'm going to skip, in the interest of time, some data where we showed that indeed macrophages do make, these toxic aldehydes, that if we make a mutation in a macrophage that mimics this mutation in the humans, that the macrophages make even more aldehyde. And so I'm going to just show you 2 more pieces of data that I think are two most important pieces of data we have. So the problem with when you only do research in humans and you look at, like the epidemiology studies that I showed you, is you can't sort out cause and effect. Are people with this mutation more likely to have active tuberculosis because they lead healthier lifestyles? Or is it because they're, our hypothesis that their macrophages make more aldehyde that kills bacteria? So this is where the power of animal models comes into play. And so what we did is we made, using two different kind of lines of mice, one that is very susceptible to TB here, that's the blue, and one that's very resistant to TB here, that's the gray. We made a mouse that has a mutation that is, the same as what we see in humans with this mutation. Okay, does this make sense? I'm just looking at Kate and Sonia because I can see your faces. So thank you for nodding. Okay, so what we did is we compared what we'll call the wild types. That means they have a functional version of ALDH2 and a mutant, which means that they have a non-functional version of ALDH2, just like the humans. And then we infected these with TB. We waited a month, and then we counted the number of bacteria in their lungs, and that's what's shown in this graph. So what we're seeing here is in this wild type mouse here, on average, they have about 10 times more bacteria in their lungs than the same mouse lacking ALDH2, so mimicking the human mutation. So what this shows is that these these mice that mimic the mutation that we see in people are protected against tuberculosis. And we see that on this strain of mouse that's very sensitive to tuberculosis. And then we see that in this strain of mouse. So you compare here and here, we see at a month after infection about a 3-fold better ability to control infection in the mice lacking ALDH2. And we don't think it's just tuberculosis. So I'll just show you one last piece of data. I was too ambitious when I made this talk. I'll just show you one last piece of data, which is that we infected with this other bacterial pathogen called Francisella. Francisella is kind of like anthrax. It's out there in the environment. Very rarely someone gets infected with it, but there's no person-to-person transmission, so we don't really worry about it except as a bioweapon. But the reason that we used it here is because it's just a really nice kind of model for something like TB that's an intracellular pathogen. And so again, we took these mice, we infected them this time with Francisella, and then we looked at their lungs. And what you can see here, just focus on this bar graph right here. This is 5 days after we infected these mice with Francisella. This is our wild type strain that has the functional version of ALDH2. And this is our mutant strain that has the non-functional version of ALDH2 that mimics the human mutation. And what we can see here now is that these mutants have 100-fold less bacteria in their lungs than the wild type. So in summary, what we think we have discovered is that one of the ways that macrophages kill bacterial pathogens like TB is by kind of taking advantage of the fact that they make these toxic aldehydes as kind of byproducts of metabolism and using those aldehydes to kill intracellular pathogens. And in humans, at some point in East Asia, there arose someone who had a mutation in this ALDH2 gene. And that actually, in some ways, is detrimental later in life because you have more of a chance to develop cancer. But the trade-off is worth it because earlier in life, you may be able to better protect yourself against bacterial infections. And so what we're working on now is for people who are wild type for this, can we find a small molecule like a drug that will inhibit ALDH2 and kind of mimic the effect of this mutation? And can we use that as a new type of drug? It would be in combination with classical antibiotics, but as a new kind of drug so that you can both kind of kill the bacteria and then target the immune system to kill the bacteria at the same time. And we're pretty optimistic about this because there are drugs that are used that already inhibit this enzyme that people use them to treat alcoholism. So that, people who don't have this mutation, if they're alcoholics, then you give them this drug, then they feel sick when they drink. And actually, it's already been shown, we've already shown that these drugs can work against TB in the mouse model. And so we're really working on trying to optimize that and trying to figure out if we can get into a clinical trial for humans. Okay, so I think we have 4 minutes left. Is there any questions? I hope that made sense.
Yeah, no, it was very good. And I really, but the last part of the alcohol and the treatment they use for alcoholism. So that was very interesting.
Thank you.
Yeah. I don't see any questions in the chat. So-
I just have a quick question or comment. There was something in a New York Times article about a drug that's used to treat alcoholism, and how it's not more widely spread, or the use isn't more widely spread. I forget what it's called. It's like, it starts with an N. Is that the type of drug that would block the enzyme?
Is it antabuse? Is that what you're thinking of?
I think I might still have it up. It's not, what is it? Naltrexone?
Oh, for alcoholism. Oh, I didn't know that was used for alcoholism. I don't know the mechanism of that one. There is a drug called antabuse that is used in like widely around the world. We don't use it that much in the US, I don't think. But I gave a talk about this in Sweden last month, and every alcoholic in Sweden apparently is put on antabuse. We don't use it that much because it's not a very good drug. It actually inhibits a lot of different things, and so it itself, there's some toxicity associated with taking it. Yeah, so I don't know. I'm not familiar with the use of what was it you said, naloxone?
Naltrexone.
Naltrexone.
I don't know if it's the same class of drugs or not. It just, I read it this morning, and so I was wondering.
Oh yeah, okay, no, this is a different mechanism. Yeah, this is a drug that would that actually targets like the addiction. I just Googled it. It targets addiction and craving. So it would be, you could use it for alcohol, or you could use it for opioids or whatever. So it's targeting that brain circuit, whereas this is actually targeting this enzyme that detoxifies ethanol. So when you drink it, you just feel terrible.
Sarah, I have one question. I'm going back to India. So is it it's such a high number? Is it because of the underdeveloped country, or is it because of the population is so high, and or the people stop completing their entire course of the medicine? What is the main reason behind that?
All of the above. A lot of cases, the population is high. The drugs are available over the counter, so people take them for a short time. They don't actually clear the infection comes back, then it's drug resistant. The other thing is that TB spreads the best in really dense urban housing that is like informal settlements is where TB really spreads the best, like a whole family living together in one room, really close to a lot of other people around as well. And so when people are living in that kind of density without modern HVAC and stuff, TB really spreads really well.
Yeah. Thank you.
Okay. Thank you for attending our BRAG meeting today. We are putting a short survey link in the chat. Please take a minute to give us feedback on today's meeting. We will also send the survey out in an email. Our next BRAG general meeting will be Tuesday, April 4th from 12 to 1. Nome Pines with Sponsor Projects Office will present on contractual clauses and indemnification. We hope you are having a great start of 2023, and we look forward to seeing you in April.
Thanks, everyone.
Thank you, Sarah.
Thanks, Sarah.
Bye.